morning. This is where we're putting our results and interpreting our results, okay? All right, so let's start from the beginning. If you have plain old nutrient agar, what are your bacteria going to look like? Look at your tri-plate of nutrient agar. What do your E. coli look like? White, and they have kind of an irregular edge. What do your salmonella look like? They're also white and have kind of an irregular edge. Your staph epidermidis, those are also white, but there's much less growth. They didn't grow very well.
This is stat. This is
If you work with bacteria a lot, you can actually see that three of them are slightly different. I've done this experiment in the past where I used Serratia, which has a red pigment, Micrococcus, which has a yellow pigment, and E. coli, which is white. So you would be able to see the difference easily. Well, for this experiment, it's different. You have unknowns, and they're all Enterobacteriaceae. So they're all going to be similar looking colonial morphologies. Also, in your microscope, the cellular morphology, they're all probably going to be pretty similar. Gram-negative single bacilli. I did notice one of the types of bacteria also kind of grows like a long chain of cells almost. So that's a little unusual. I have seen it before, but that's something you need to mention alongside your little gram-negative bacilli. It can also produce those long strings. So you write down your colonial morphology in this chart or wherever. You write down your cellular morphology. And now we're going to compare the plates. With the plates, we should be able to rule out what it's not from the known bacteria. And if it resembles one of the known bacteria, that might help you confirm that it's the same as the known. So, for example, I wish I probably have a set of plates to show this. Okay, the EMB agar over here stands for eosin methylene blue. The eosin and methylene dyes harm gram-positive. So it selects for gram-negative and against gram-positive. So do you think your S. epi is going to grow on it? Absolutely not. It's gram-positive. The other two can grow on it because they're gram-negative. It specifically selects for enteric bacteria. And there's lactose in it, which supports some enteric bacteria like your fecal coliforms. Who is a fecal coliform? E. coli. When E. coli grows, it's selected for gram-negative enteric, so E. coli will grow. But when E. coli grows, it's going to be differentiated from other gram-negative enterics because E. coli will take up the eosin and the methylene blue, and the colonies themselves will turn metallic shiny green. So somebody who's got metallic shiny green, please hold it up and open your plate so the other people can see. Does anybody see green, shiny green? If your unknown looks metallic shiny green, like your E. coli on your tri-plate, you probably have E. coli as your unknown. Does that make sense to anybody?
You said that.
Okay. Now, that's what I wrote over here. For your EMB agar, shiny green would indicate E. coli. What if it's not shiny green, but it's black or dark purple colonies? That indicates that they are taking up the lactose in the agar, and when they take up the lactose, they take up the dyes. That's what turns them dark black or dark purple. That means they're strong lactose fermenters. They love lactose. They're taking it up out of the agar, and when they do, they turn color. E. coli is distinctive because it produces the metallic green shiny colonies, and it's a lactose fermenter, a very strong one. But other enteric bacteria that ferment lactose will also take up the lactose and the dyes. They just don't turn shiny green. So you would know that I do have a strong lactose fermenter. It just doesn't happen to be E. coli. So there's a clue there. You've got a strong lactose fermenter, or you might have E. coli. If you didn't get a color change, it's just kind of a black color on the purple agar, then it's a non-lactose fermenter. Lactose fermenters are considered friendly coliforms, and non-lactose fermenters are considered not friendly to humans. Okay, let's go back to the next one. Your McConkey agar plate. This plate is also got agar in it that inhibits gram-positive bacteria. So SFE cannot grow. It selects 4 gram-positive, it selects 4 gram-negative enteric bacteria because it also has lactose in it, and a lot of them like it. And it's probably got sugars in it that they like. So, your McConkey agar, your E. coli should grow because it is Enterobacteriaceae, but if it's a strong lactose fermenter, you're going to get a strong color change. It'll turn milky pink because it's taking up the lactose, and when it does, it also takes up the pink dye. So the colonies themselves turn like a milky pink, like the little bunny Nestle Quick, the strawberry flavor. That's what it looks like.
It's pretty.
Salmonella is Enterobacteriaceae, so it will grow on McConkey agar, but it's an unfriendly, non-lactose fermenter. Salmonella, no, we don't. E. coli, yeah, we have E. coli. Salmonella, we don't have salmonella. It's pathogenic to us. Chickens have salmonella. So it's a non-lactose fermenter, unfriendly. You'll get growth of it, but it won't have a color change. It'll just be sort of a dull color. So, in summary, on your McConkey agar, only gram-negative enterics will grow. And when I say enterics, in this case, I mean Enterobacteriaceae, because you do have enteric Enterococcus. That's a different division, a different section. So here when I say enterics, I'm specifically referring to Enterobacteriaceae. Okay, enterics will grow, and if they're lactose fermenters, you get a strong color change to milky pink, or at least some milky pink. You might have a weaker lactose fermenter, you got some pink but not a whole lot. But if there's no color change, it's an unfriendly non-fermenter. Shigella salmonella agar is specifically made, and they don't really use it that much anymore, they have other agars they use instead. But it's specifically made to show you when you have salmonella or shigella, which are unfriendly. Shigella probably come from fish poop, salmonella from chicken poop. These are harmful, cause food poisoning for us. They are enteric flora, but not for us, and they can be pathogenic if we ingest them. So, the Shigella salmonella agar inhibits gram-positives and selects for gram-negatives. But if your gram-negative is salmonella or Shigella, which are non-lactose fermenters, instead of getting lactose fermentation, you're going to get the black precipitate.
Mm-hmm.
Who got the black precipitate? They did. Crystal, would you hold up? Yeah, hold up your plate so they can see how black it is. See how black it is? Now, some of the tests we do in the API strip are going to be the same test that you did on your plate, and it will turn black. Another test will be for lactose or whatever, so we do some of the same tests on the API strip. But I just like it on the agar because you can see it. So when it comes to your Salmonella Shigella agar, if you got the black precipitate, it matches your Salmonella plate, the tri-plate where the Salmonella is. You know you've got Salmonella. Very few bacteria make that black precipitate. If it's growing, but you don't have the black precipitate, maybe it's E. coli or something else. Last one.
What about the coli? Does it grow in Salmonella?
I think it does. And you'll get some milky pink. So Salmonella Shigella agar also has lactose in it. So if you get the pink colonies, it's a lactose fermenter. Salmonella is a non-lactose fermenter, turns black. So if it's black, it's a non-fermenter, unfriendly. If it turns pink, it's a fermenter friendly, could be E. coli or some other gram-negative bacillus like E. coli. The last agar is your mannitol salt agar. This one selects for gram-positive Staphylococcus. It's the salt. Remember that. It'll be on your test. Staphylococcus likes salt. Not an extreme salt lover, just mild salt, like on your skin. Staphylococcus epidermidis, all over your skin. Micrococcus luteus, Micrococcus roseus, they could be there too. Staph aureus can be there. They're all Staphylococcus. Staphylococcus likes salt, so we add salt to the agar so they'll grow. Gram-negatives don't like salt, they won't grow. You're covered with Staphylococcus. You go for a jog, you start sweating. You come home, you're all sweaty, your dog runs over and starts licking your leg. Because you taste salty. He's licking the salt. He likes the taste of the salt in your sweat. Staphylococcus like your salty skin too. So that's why they grow so well on our skin. Not so well on nutrient agar because there's no salt in it. There's just a little bit. The mannitol salt agar has salt added to promote the growth of gram-positive Staphylococcus. Not just gram-positive, specifically gram-positive Staphylococcus. The mannitol is a sugar. If the Staphylococcus can ferment mannitol, it will take up the mannitol, digest it, and excrete acid waste, and the acid waste will turn the agar yellow. Pathogenic bacteria, pathogenic Staphylococcus like Staph aureus, turn the agar yellow, they're pathogenic. If it doesn't turn the agar yellow, it just stays pink, and it grows, it's Staph epi, non-pathogenic. Now, your gram-negatives should not have grown. They may grow a little bit. The salmonella actually grows a little bit, might turn your agar a little bit yellow, but you can disregard that. Just put no growth. But if you think you have salmonella, and it did turn your mannitol salt agar a little bit yellow, that happens. That might also help you identify it. So then at the bottom of this document, I wrote a few little sentences to summarize. The EMB, the MacConkey, and the SS agars all select for gram-negative Enterobacteriaceae. And they differentiate lactose fermenters, considered friendly normal flora, from non-lactose fermenters, which are considered unfriendly. The EMB agar specifically can be used to identify or rule out E. coli. If you're unknown as shiny green, like your E. coli on the triplate of EMB agar, probably have E. coli. If it's not shiny green, it's not E. coli. On your MacConkey agar, this also determines lactose fermenters and non-lactose fermenters. If you get a strong milky pink, it's a lactose fermenter. If you don't get that color change, it's a non-lactose fermenter. If your E. coli known turns milky pink and your unknown on the MacConkey agar also turned milky pink, it could be E. coli. Especially if you also have the shiny green on your EMB agar. Get it? It might be salmonella, it's just growing, but it's not getting a color change.
Also from here.
The SS agar is very specific for salmonella. If it came out black, it's salmonella. Proteus, also, if it came out black, it could be Proteus. I doubt highly that the lab techs would have given you Proteus. So it's probably salmonella. And the last one, mannitol salt agar, is just to show you that what your unknown is not gram-positive. You do not have gram-positive Staphylococcus. Your unknown
Because there was no
can't grow
growth.
on the mannitol salt agar.
No growth.
Did that help you understand